Um, all right, let's get started. Welcome, everybody. Good afternoon and good evening in case you're coming from Europe. Um, Welcome to our third webinar of the year. And today we'll be focusing on our mouse models that can support cancer research. And it's really my great pleasure to have Dr. Pradeep Kumar Sasisaran to do the presentation for us today. He is our Vice President of Business Development. Dr. Sasisaran has a PhD in molecular and cellular medicine from University of Oxford. He traveled extensively during his postdoc period. He was a Fulbright Scholar at Harvard University, a Daniel Turnberg Fellow at the Hebrew University of Jerusalem. He also was a Amble Fellow at Sorbonne University. By doing so, he gained a rich international scholarship and research experience in inflammation and immunology. Prior to joining Gempharmatech, Dr. Sasisaran has established his own independent research lab at Xi'an Liverpool University, during which time he was very active and successful in doing research in aging and inflammation and arthritis. Today, Dr. Sasisaran will share the knowledge we have gained in developing new mouse models for research in immuno-oncology and immunotherapy. Without further ado, Pradeep, the stage is yours.
Well, thank you very much, Jing. Thank you very much for that wonderful introduction. So today we're actually going to have a brief introduction. Apologies, I'm going to do it because our CEO, Dr. Mark Moore, had to be called on urgent business. So you're stuck with me for 40 minutes, but I'll make it worthwhile. So I'm going to have a small introduction, probably 10 minutes on who we are and what we do. And then we're going to talk about the actual topic, the actual main course for the day, which is about novel models to avoid pitfalls in immunotherapy. And we're going to show you some very exciting data from Gempharmatech to see what we think, how we can actually add or decrease, I should say, the failure rate in preclinical research, and how we can help researchers avoid the classical pitfalls that we see. So, what about Gempharmatech at first? Who are we? We're really a one-stop solution for preclinical mouse research, and that sums us up very well. Before I start, I'm going to have a quick disclaimer here, and then we're going to go to our company history. So the company history itself, we see that Gempharmatech was originally created by Professor Dr. Gao Zhang in China in around 2002. And ever since then, we have some very prestigious landmarks. And in 2006, we were actually AAA LAC accredited, which for those who don't know is kind of the gold standard that shows mouse facilities and our research capabilities are up to international par. And we've actually had that accreditation now for 14 years. And along the way, we also had some major landmarks, including 2011, we were included in the International Mouse Phenotyping Consortium. So that was an international network working together to create knockout mice and phenotyping different knockout strains. And in 2014, we actually was a rapid growth year for us where we established our research facility and research building. And in 2013, a year prior to this, we had established a lot of platforms, and we really actually started working with CRISPR-Cas9 in this year, in these 2 years, and we become very renowned in for this technology in creating these mice, and we've become established, and the production rates themselves has increased exponentially. And in 2017, we actually formed our company and named it Gempharmatech. And also we capitalized on 2 rounds of financing ever since then. And now we have opened branches in 3 or 4 now different parts in China. And we also excitingly launched our US branch where we actually now have housed over 6,000 research models in the US, and we also serve Europe as well. And these numbers of models are ever increasing. So it's exciting times for Gempharmatech, and it's a global expansion. And what we really do is split into 4 or 5 different kind of main categories. We have the classical mouse model resources where we make conditional knockouts and knockout mice and I'll talk about this exciting platform soon. We have our humanized mice, the immunodeficient mice, and our germ-free mice as well. And we also do preclinical efficacy and phenotyping. We have a lot of disease models available. We also do preclinical drug discovery as well. And we also specialize in mouse phenotyping, which is also it goes in hand with our mouse breeding preservation services as well. So we can do the classical things like cryopreservation. And what's exciting is that we never say no to any researcher and their ideas and what kind of dreams they come up with in the science field. So we have a model customization service that's second to none in terms of how we can create the custom models using CRISPR-Cas9, even old school ES technology, and also how we can produce these new transgenics to help the researchers. And now, being a very global company, we are experts in import and exports of these models as well. So no need to worry about delivery hurdles and so forth. We take care of it. So I'm going to touch upon the platforms of Gempharmatech. Before that, I'd like to show you that we have world-class facilities at our locations. And as I mentioned, we have triple A LAC accreditation. We also have the ICLAS Microbial Reference Laboratory accreditation as well. And our capabilities are ever expanding. So it's exciting times. And what we're very proud of is our ambitious and knock out all projects. We call it the KOAP project, where we have set the target and goal of floxing all 22,951 protein coding genes. Our capacity now is around 5,000 lines per year. We flox majority of these genes up to nearly 8,000 already. We also have conventional knockout strains available as well. So this is a very exciting, ambitious plan, which is actually coming together very well. And we have a lot of orders. So all of you scientists out there, please reach out to us if you have a favorite knockout line, or if you want to flock something. We're here to help you, and we've probably got it already as well. So if you talk about conditional knockouts, you need a Cree as well, right? We got you covered. We have Crees across the physiological system, across cell types and tissues as well, from nervous system to immune system. The last count is 192 different strains of Cree. It probably increased by now. But yeah, so if you want flox mice with Cree, we can serve you as well. So that's our knockout and Cree platform. And one thing we're also very proud of is our preclinical discovery platform, where we can help the industrial clients, even from big pharma to small and medium tech, biotech, or CRO research companies, whoever you are, to actually do PK studies, look at discovery platforms in different disease models. And we're expert in many things, but we really have nailed down the immuno checkpoint inhibitors and drugs. And these are some of the examples of what we've done in the past. And going forward, a third platform I want to highlight is our microbiome and germ-free platform, where we have the classical genetic engineered mice in the germ-free kind of arena and background. We also have the ability now, and what's exciting is to actually, if one of you says, I want to study rheumatoid disease or cancer disease, we have a floral transplantation service where we can actually use and take the flora from patients and actually put it in these germ-free mice and give you some interesting data if you wish to. And we can also combine certain aspects like a humanized mice into a germ-free kind of background, and we can see how these are going as well. And excitingly, what we're also building is a capability of looking at wild type mice. When I mean wild type, I don't mean the classical C57, I mean the wild type outside the laboratory. And this is a huge growing area in the microbiome research. And it'd be interesting to see where we can go on from here. And I also want to talk about immunodeficiency models. We have new immunodeficiency models coming out all the time, which are highly specific, and we have a lot of success in terms of how low the immune system is actually working. And we have now built more from our from other lines as well, and incorporating humanized backgrounds in these mice as well. So that's what we do. As you can see, I highlighted 4 platforms, and we really are. Hopefully, I can summarize and tell you that we're a one-stop solution for all your preclinical research needs. So please do come to us and talk to us, and we're here to help you and serve you, and we probably got you covered in all aspects. So that's the introduction to Gempharmatech. What I really now want to talk about is getting on to how we can use our novel models to avoid the pitfalls in immunotherapy. We are very excited with some of the data we have now in this very small aspect, and we're going to share some data and models what we're thinking about and how we can actually help our academics and also our industrial collaborators in drug discovery. Now, I might be preaching to the choir. I know there's a lot of experts here, but as we know that cancer immunotherapy has gone on, has rapidly expanded in the last 30 years or so. And what we see is a lot of drugs now coming out looking at checkpoint inhibitors in cancer immunotherapy. And there's been a lot of success and a lot of revenue actually generated. And as we know, if we have drug success, beforehand we have academic success, and some of these cancer immunotherapy drugs have reached high impact factor papers. And we know why. One reason why this kind of avenue is working is because the antibody molecule is highly specific target and it works. It's worked for many diseases and it's working for cancer immunotherapy. So it's a good sign that it's working itself. But one of the things that many don't know, and most of you might know, is that the early preclinical discovery costs are going through the roof. They're ever expanding, and it's getting even more expensive to make drugs. And this huge cost at the early stage is actually crippling some people, or is actually making others merge together, trying to get money together to see how we can actually work things out or actually discover better. And one of the reasons is that if you can see these figures, if you start with 80,000 patents of some kind of a novel compound, by the time it gets to preclinical research, there's a huge drop. And then from the preclinical research stage, uh, and from that research going on to the human stage of phase 1, you see a further decrease. So what we're trying to do is answer the question in our own ways, how we can help scientists in this early stage, how we can get these drugs validated very well and then again not fail in the phase 1. And one of the reasons why scientists and many industrial collaborators say is that the mouse model is not working very well here. What we see in the mouse model is not mimicked or recapitulated in the human kind of disease area in the humans itself, and what we can do about it. So one of the problems is a lot of scientists say, well, the mouse is not a very good model. We argue it is because a lot of science is highly dependent on a mouse and it's been validated robustly in these systems. But we also say that it can be improved. One way we're improving this from Gempharmatech is using our unique capabilities in humanized mouse models. And what do we mean by humanized mouse models? Well, if a scientist or industrial collaborator says, oh, we need to make this mouse model better to fit the human disease. So we do 2 things. We actually can make a gene edited mouse model, which we call GEM, which is essentially a human target, a humanized human target plus the mouse immune system. Or we can take it one step further. We can have an immune reconstituted model, which is the human, for example, we take human PMBCs and also human stem cells, and we can engraft this on an NCG background, for example. Hence, we have a human target plus a human immune system. So this is a way we talk about humanized mice, and we can study cancer immunotherapies very efficiently. Um, so we can have this approach, and many are actually using this approach. But the problem we have is twofold. We have 2 questions always arising. We say which humanized mouse background is the better for immunotherapy, And we also have the question is which humanized mouse background is more sensitive to toxicity of immunotherapies. So what background, or if a single background of a mouse affects immunotherapy, and can we study toxicology, which is actually a common, or I shouldn't say common, but occurs in many drug discovery phases, if we can actually study toxicology for immunotherapies very well. So I'm going to answer the first question at first. So we have example of the TIGIT immune molecule, which is actually being used for cancer immunotherapy. Now, if we look at this data, what you see is you have a B6 background mouse, which a human PD1 humanized gene with a humanized TIGIT gene as well. And in this experiment, we're using this mouse with the MC39 cancer line to induce cancer. And what you see is, you see here we're measuring tumor volume. And if we give an anti-human TIGIT drug, right, which is meant to be good in reducing cancer, in this model, in this mouse background, we see no difference compared to the control. So this is very interesting. So this may see a researcher might see this and say, oh, the human, the human TIGIT is antibody doesn't work very well in this cancer model. So what's going on? Well, what is going on? Because the mouse data is not showing much, but if you see the latest breakthroughs, and we have Roche actually have a novel anti-TIGIT antibody having an FDA breakthrough therapy designation. We have other companies actually looking to launch this and being approved in clinical trials. So there's a huge disparity between the human data and what happened in the preclinical data. In this example, in this immunotherapy, we think that the B6 background doesn't work very well. And we saw this disparity some time ago, and we wanted to research what's the difference between the B6 background and the BALB-C background. Before I get to that difference, I want to talk to you about the study outline we did and what kind of experiments we actually did to answer this question. So we took a BALB-C mouse and also a B6 mouse. For the BALB-C, we used classical cell lines for each background. So this is a C26 cancer-inducing cell line for BALB-C. And for the BLAK6, we used the MC38 cancer cell line. And we actually allowed the tumor bearing to occur for 2 weeks, and then we grouped them in terms of tumor volume. Then we gave the respective drugs for 3 to 4 weeks, and we measured body weight and tumor volume. We also we stopped observation and then we let them go for 1 more week, and we looked at endpoint readouts, which is again body weight, tumor volume, and we looked at blood tissue collection and also profiling the cells as well for lymphocytes analysis. What we saw is very interesting. So you saw that data prior before in the interesting slide on the B6 background with the humanized PD1 and humanized TIGIT mouse with the MC39 cancer. You saw no difference when we used the human TIGIT antibody against control. So in the B6 background, it looks like if this drug is not working, right? There's no difference. But in the BALB-C background, same humanized genes, in a cancer model with the same drug, you can see that the drug actually has a reduced tumor volume. So this shows that the drug is actually working very well, and what we see in the clinical trial launched as well. So this shows us that the BALB-C background for this kind of TIGIT antibody, and it might be better to use. So we now have a difference in background. So we can see that the BALB-C mice might be better and recommended. So we actually went on to look at this even further. We didn't want to take any chances, being robust scientists. So we looked at another anti-TIGIT antibody from Genentech and Roche as well. And we looked and we saw the same difference compared to IgE control. You see tumor volume decrease with this other drug. And we also went one step further and asked what the mechanism could be. and not getting into too much detail, we used the drug and we had a mutation, the FC portion, to abolish the effects of antibody-dependent cellular toxicity, which is ADCC. And we saw the difference not working in this. So this shows that the ADCC effect is actually very important in this background and for this drug effect to work. So we saw the BALB/c background perform very well with two different human TIGIT antibodies. And we also saw that there's a difference in tumor infiltrating lymphocytes analysis and immunophenotyping the cells. Let me walk you through this data. If you look at the BALB/c background here, you saw when we looked at the NK cell, there's not much of a difference. So the the B6 background is where the drug did not work. And we saw here the CD3 positive T cell population increased slightly in the B6 background, and Treg increased as well. But in the BALB/c background, where the drug did work and showed preclinical efficacy, what you saw is actually a massive increase in the NK cell population, no change in the CD3 T cell population, and also a reduction in the Treg population. So what we're seeing is a different immunophenotyping and background in terms of background. So in the BALB/c, you see a difference in immune cell function and expression as well. So that's very interesting to see. So we went on actually to look at the literature to see if others are seeing this effect. And we are very pleased to see that others are seeing this effect. And this group here in the Journal of Immunology actually used two different cell lines for cancer inducement. And they looked at tumor volume in the BALB/c background, and they saw exactly what we saw. That in the BALB/c background, when you target TIGIT with an anti-TIGIT antibody, you have a decrease in the tumor volume. So that shows that for this immunotherapy, the BALB/c background is better. And they also saw the same effect and said that the effect of this background and why it's working is dependent on ADCC. So there was a academic robust data showing that our data and the BALB/c background was validated and works very well as well. And also the academic group in this journal article saw again differences in kind of phenotyping the cells. And just like us, they saw a difference in the FOXP3 positive Tregs with the drug. And they also saw a difference with the drug when the CD8 T cell depletion occurred, and also NK cell depletion occurring as well. So this shows us that the BALB/c background is better for human TIGIT analysis, and this might be due to the difference in antibody-dependent cellular cytotoxicity, and also the difference in immuno cells and their profile as well. So this was very interesting data, and some data that some of you can go along and look at, and we are all more than happy to help you in your journey and actually address these things as well together. So in summary, what we think is the question of which humanized mouse background might be better for certain immunotherapies, especially the human TIGIT therapy, we'll say is the BALB/c. By no means are we saying the BALB/c is the be all and end all and that you should use all the experiments, but certainly background of the humanized mouse background is important when you're considering immunotherapy research. Now we're getting on to the second question is which humanized mouse background is more sensitive to toxicology of immunotherapies? Before we get on to that, what is toxicology of toxicity of immunotherapies and why is it important? Well, you have this term called immuno-related adverse effects, and it occurs more than we think in many different areas and physiology when an anti-immunotherapy is given. So it can occur in the renal failure, or it can actually it can be minute as a skin rash, or it can be very serious as colitis and diarrhea, or you can have neuromuscular effects. So immune-related adverse effects are very kind of common in certain areas and should be looked at. But how can we study this effectively? If we have a drug and we think it's going to work, how can we look at the side effects it might induce? And that's a very tricky question. And before we get to the data, for example, if you have a anti-CTLA4 drug, CTLA4 is a classical immune molecule that's targeted. And when you target it with an anti-CTLA4 drug, what you see in the patient is that when you look at distribution grade of these adverse effects, you can see that the patients suffer a lot in terms of skin and also in terms of high grade in the GI tract. So if we're going to take this example, we wanted to research on this anti-CTLA4 and what kind of toxicity effects can we study and why we sometimes not seeing toxicity effects in certain backgrounds and in other backgrounds we do. So if we first of all look at the human example, remember one of the classical questions like what we see in the humans is not happening in the mice. Why? So we looked at this disease and this anti-CTLA4 drug called Yerway, and we looked at if we look at the human disease published here, um, this patient here got a dermatitis as a adverse effect, and also he went on to get hepatitis as well. And this was very intriguing to see, and we wanted to delve deeper into this. So we looked at first adult B6 mice and what happens if we give a anti-CTLA4 antibody. And what we saw in the literature is that when you look at the B6 mice, the adult mice showed very mild toxicity, if not much. So if you use a big B6 background and you have a humanized gene in this background and for CTLA4, and you use an anti-PD-1 antibody, which is for cancer therapy, and also what you see is mild toxicity effects. So it doesn't recapitulate the kind of adverse effects you see in the humans, as I pointed out. So I should say this has an anti-PD-1 and anti-CTLA4 drug as well. But some of you clever people out there will say, oh, use adult mice. Why can't you use young mice? Yes, you're right. If we use young mice, they do show adverse effects to CTLA4 antibody. But the problem is there's a high death rate in young mice when you look at this antibody. So the mice actually are dying very soon, and you need a lot of N number, an increased population to study the effects very rapidly and efficiently. And also, it's not very ethical to use a huge bunch of mice knowing that they're going to die. And what's more of a problem is that if you look at the human disease, it's manifested in the adult population, not the young. So using a young B6 mice because it's toxicity effects of the antibody might show is not very wise because the human disease manifested in the older population. So how did Gempharmatech get around this? So before I get to that, I'm going to introduce the experiment itself at first. We use 6 to 8 week at time points here, and we use the BALB mice with human PD-1 humanized gene and the humanized CTLA4 gene. And then we compared that to the B6 background with the same genes for PD-1 and CTLA4 humanized. And then what we did was give anti-CTLA4, which is immunotherapy, and also anti-PD-1 immunotherapy as well. And we left it for 4 weeks. We looked at survival curve, body weight, and we observed them as well. And after stopping observation, we also looked at blood analysis and also H&E analysis as well. And the data was fascinating. If you look at those mice, the humanized mice on the B6 background with these two humanized PD-1 anti-drug and humanized anti-CTLA drug, what we saw is in the B6 background, survival actually was 100%, and there was no difference between control and drug. This shows that the mild toxicology that the research group reported was also seen in our mice. So the toxic effects of the drugs seen in human is not very well established or mimicked in the B6 background. Whereas if we use the BALB-C background for these drugs, we saw survival rates decrease, showing that the toxic effects seen in the human population might be more mimicked or recapitulated better in the BALB-C background. So that's the survival rate data. And when we looked at observed the mice, what we saw was a huge difference in the BALB-C background compared to the B6 background. The biopsy background clearly displayed joint inflammation, adverse effect to the drug, and also lost a lot of weight and were just generally worse off compared to the B6 background. So observation wise and survival curve wise, and in terms of joint inflammation, we saw that the biopsy background had a more effect and was better to look at toxicity of these drugs. We didn't stop there. We actually looked at the internal organs, and what we saw in the biopsy background that these mice given the drugs had actually they had intestinal bleeding and also enlarged organs here compared to the B6 background. So the toxicity effect was occurring internally as well. And we also looked at other classical markers, and what we saw was LDH and also CK increase in the biopsy background, showing that there's actually severe heart damage or cardiac damage caused in the biopsy background after treatment of these drugs, so mimicking the toxicity effects in humans. And also when we looked at H&E stains and we saw inflammatory lesions occur in murine hearts in the biopsy background treated with the drugs compared to the B6 background. And finally, this intrigued us. We thought, oh, if this is happening in cardiac tissue, what happens um, in other organs and tissues? And what we saw is that in multiple organs, the liver, lung, kidney, the saliva glands, intestines, we saw huge inflammatory lesions when the drug was given against CTLA4 and PDA1 in the BALB-C background humanized mice compared to the B6 background. So this was very interesting data, and hopefully we wanted to show the world that if you look and take a humanized mouse and target CTLA4 and human PD1 in a biopsy background, it's actually mimics the toxicology effects very well. But the question is that we use one antibody. What if we use a different antibody? And here we used an antibody against 41BB, which is a also CD137. And we used the antibody and we saw the similar effects. This time we saw severe liver damage occurring in the biopsy background measured by ATLA um, measurements. And you can see these graphs look similar, but look at the levels here. When you give the drug and without the drug, you see the biopsy background has a huge difference in ATL levels compared to the B6 level as well, showing that the toxic effects of the drug may be better studied in the biopsy background. And we went on in collaboration with Cryo Biosciences to look at the tumor volume in the drugs against 41BB. So we used the BALB-C background with humanized CD137, and we targeted with a 41BB antibody, which targets CD137. And what we saw is with the drug in the BALB-C background, we saw a decrease in tumor volume, just like the earlier studies I showed you in the first question. And we saw this effect working very well in the biopsy background, and we also saw a difference in body weight as well. So in summary, question one was answered, which background do you think is better for certain immunotherapies, and that's BALB-C. And we saw that for CD137 and the PD1 drugs and also the CTLA4 drugs, if you look at toxicity and look at these studies, we also recommend the BALB-C background as well. But I must say, this does not mean that the BALB-C background is the best for all immunotherapies. we're trying to point out that you must ask the question, and you can talk to us as well in terms of what molecule am I studying, what drug am I studying, which humanized background am I using, and which humanized genes am I using, and that might have an effect on the endpoint readout. And we've become world experts at this, and we can actually advise you very well and guide you through this maze, as it were. But it's important because some of you might not be seeing the effect, and that might not be because of the drug, but it might be because of the background. So I don't want you to go away saying biopsy, biopsy, biopsy. Gempharmatech is not that biased, as I explained. We know there's a lot of research, and the B6 background actually shows some good effects in different drugs and different conditions. So that's something to remember as well. And what we did is actually considered all our customers and all the results, and we now humanize animal mouse range here. We actually have single targets for classical markers like PD1, PD ligand, and so forth, but we went on to have double targets and also triple targets on both backgrounds. So if you're a researcher and you want to look at human PD1 with human PDA ligand and a humanized CTLA4, we've got that mouse. And it seems extreme, but we can actually now very rapidly make different humanized mice together, and we can actually customize these mice as well. So you name it, you want single target, double target, triple target on whatever background, we can help you as well. And also, I want to leave you with this slide as well to say that we're not a one trick pony. As I showed you, we're not all about immunotherapy. This is just a tip of the iceberg of what we can do. I showed you 4 different platforms, but we can use those platforms and go across all disease ranges from diabetes to cardiovascular disease to autoimmune diseases. And then we can humanize these mice. We can put a germ-free background if you want. We can use any background. We can actually make a knockout mice for you and also put it in these disease models as well. And then on top of that, we can also study pharmacological effects as well in our preclinical discovery platforms as well. So we are really a one-stop solution. We hope you can reach out to us. Just have a chat to us. We have a friendly team in the US, in Europe, and across the world. And you can just ask us any questions. We're not here to sell things or make you accept things, but we're really here as scientists. Most of us have PhDs, and we're here to help you, and we're here to actually share our data and see how we can actually progress science together. So I want to thank you all very much for your attention, and I want you to leave. I'm going to leave you guys with this slide. As I said, reach out to us. Our best email address is globalservice@Gempharmatech, and we'll hope to hear from you. And again, thank you very much for your attention.
Great talk. Thank you so much, Pradeep. So,
We'll take some questions now, Jing.
yeah, I'm keeping an eye in the chat box as well as the Q&A. We did receive a couple of questions. I'll start with the first one that we got. Um, this participant asked about the ticket model, saying, you showed a ticket with a PD1. So he's wondering, or she is wondering, does Gempharmatech have the single ticket humanized animal? Do you have it on both backgrounds, as you were saying?
Oh, yes, we got you covered, participant. We have the ticket here, as you say, on both backgrounds, on a single target, on the B6 and a BALB-C background. And we went on, if you want even more, we have it combined with other humanized genes as well. And they're ready to go, and they're ready to ship, and we're here for you.
All right. The second question I saw also pretty simple. So you mentioned that you opened operation in the States, in the United States. So with that, how long can we expect mice to come to us?
So if you're based in the US, it really depends what you want. Well, I just told you that we ship 6,000 mice models in. So if one of those 6,000, and by the way, that's ever increasing list and number. So if you have it in the States on the shelf, we can expect it within a month or 3 months, depending on the model. As also, if you want a fully customized mouse, you can expect that well globally within 3 months, if not less. If we have it in our main headquarters and it's on the shelf, you can even expected lesser if you're ready to purchase and we're ready to send. But also you have to remember, if it's a fully customized mice, it might take up to 6 months for us to make and generate the mice and ship it to you, if not longer, if it needs breeding and crossbreeding.
All right. So actually this can lead to the next question. Um, the question is, you mentioned immune deficient mouse. So can you do humanization with immune deficient mice?
Yes, that's a good question. Yes, we can. So we can get a human deficient mouse and actually humanize the genes and kind of implant them, if that's the word. So we have a humanized gene with a humanized immune system as well. So that'd be very interesting study and look at those genes. And also you can tell us if you also want specialized humanized immunodeficient mice, for example, with cytokines like IL-15, and then on top of that have a humanized gene as well. We can do that as well. So the world's your oyster in terms of Gempharmatech. You tell us, we do it.
So actually, the same participant had a second part of the question. Do you have a list of your humanized mice on the immune deficient background?
Yes, we do. You can find that on our website, and also email us, and we're more than happy to go through the list with you, and also recommend what's good for your needs.
Great. I'm coming to the last question I saw in the chat boxes. It says, can you generate a humanized mouse model and then do drug testing with our compounds?
Yes, we can. I'm sounding like Obama, huh? Yes, we can. We can because we can. Because we have the facilities and ability to do so. So if we have a humanized mouse, we make that mouse for you. We can keep it in-house, and you can send us the compound from anywhere in the world, and we can do the testing on that humanized mouse. And we can actually use that humanized mouse compound on a disease model as well. So we can take it one step further as well.
Great. I think this is a question like reassuring to make sure what we have mentioned can be applied to customers overseas, not just within China. So with that, we finished all the questions that we have received. If there is more question, everyone is welcome to email us. As Pradeep said, the global service at Gempharmatech is the best way to reach us, and you can also use the phone number if you're in the States or in Europe to reach out to us. So with that, we're going to close today's webinar. Again, thank you so much, Pradeep, and thanks to everybody who joined us today. And we'll be seeing you next time when we do our May webinar. Thanks.
Thank you. Bye.